Okay, so now let's talk about regulation of genes in eukaryotes. And so we're just going to talk about the different levels of regulation and the availability of genes for transcription. So in eukaryotes, these are going to be different than prokaryotes. Bacteria, you're going to have basically activators and repressors that are going to control the availability of an operon for transcription into functional genes. And then with eukaryotes, we're going to have restrictive transcription based on 4 key differences. So in eukaryotes, just simply having access to the promoter areas are going to be restricted. And this is by the structure of the actual chromatin. And also in eukaryotes, you're going to have positive mechanisms, which is going to be the dominant regulator in all systems. Eukaryotes are going to have much larger, more complex regulatory proteins, and we have organelles. So we have a nucleus, so that's going to be separate from our cytoplasm. So the site of transcription is separate from the site of translation. Now, another thing to keep in mind is that we have chromatin condensation. So condensed chromatin is heterochromatin. And this is transcriptionally inactive. Chromatin are wound so tightly that you cannot get access to the regions within the heterochromatin. And so condensed DNA is strongly going to suppress or repress transcription. And less condensed chromatin is euchromatin. Now, active chromatin, transcriptionally active chromatin, the euchromatin have 3 different things that make them active. One is the positioning of those nucleosomes. So the presence of your histones and the different histone variants, as well as different covalent modifications in these structures. So all 3 of these, the nucleosome positioning, covalent modifications, and histone modifications, are all referred to as chromatin remodeling. So we do know we have a core of histones that are made up of histone proteins 2A, 2B, 3, and 4. All of these can be modified. They can be methylated on lysine or arginine residues, acetylated, ubiquitated, and so oftentimes the modifications of histones occur when there is some sort of damage to the cell. So acetylation, ubiquitation is going to be when there is damage. For the core histones, they're going to have a central domain, and this is going to be involved in the histone protein interactions. This is also where the core histone proteins are going to wrap around the DNA. And then there's also lysine-rich areas, and these are in the amino-terminal domain of the exterior of the nucleosome particle. So we have our HATs, or our histone acetyltransferases, and these are going to add acetylate groups to our lysine residues. So here you have your acetyl-CoA, you have your histone with our lysine residue shown here, and then this acetyl group is going to be added to the histone, and acetylated histones are going to be more transcriptionally active. Now we have 2 types of HATs. We have our type B, and this is going to acetylate in the cytosol before the histones go back into the nucleus. And so we have our A types, and this is going to further acetylate in the nucleus. And so the type A hats are going to add to sites that are being activated for transcription. So these additional acetylation sites are going to be on our proteins 3 and proteins 4. We're also going to have deacetylases. Deacetylases are going to reduce acetylation when transcription is no longer required. So when you're going to shut down the production of the protein, you can deacetylate the areas. Now, histone 3 and histone 4 acetylation can also lead to reduction in the affinity of the nucleosome for the DNA. So what that means is that your acetylation of 3 and 4 and so this is going to mean it opens up. So remember, our nucleosome is going to be our histones wrapped around our DNA. And so the wrapping of this is to compact the DNA. In order to expose an area for transcription, you have to unwind those areas. You have to have your nucleosome dissociate from the DNA so that the region of the DNA becomes available for transcription. And this is done by those type A histone acetyltransferases in the nucleus that add acetyl groups to histone 3 and histone 4. Now, another thing that's important is methylation of DNA. We've talked about methylation of DNA. And so our methylation of DNA is going to be on cytosine. And this is going to produce these GP-rich sequences that are referred to as GP islands. These typically are going to be regulatory, and so they're found near promoters. And of course, methylation is always going to reduce the transcription rate of genes in eukaryotes. So methylation of DNA is a very I would say it's almost permanent. It's hard to reverse methylation of DNA. Now, histone modification is reversible. Acetylation is reversible. Methylation of histones are reversible. But once you get to the actual DNA molecule, methylation is not going to be as easily reversed. And so this is going to be a way to do those epigenetic gene expression regulations. An example is our globulin genes that are required in our erythrocytes. They are going to be methylated in all non-erythroid cells, for example. Another complication of methylation of actual DNA is when you have diseases associated with this. So remember, methylation of DNA is hard to reverse. This is an epigenetic mechanism to make sure your neurons are neurons and make sure your gut epithelial cells function as gut epithelial cells. And so when you have something called genomic imprinting, this means that the process of silencing a gene occurs during the formation of eggs and sperm. So this is something, genomic imprinting is passed on, just like methylation of your liver cells. When they make a new hepatocyte, that methylation is passed on. But genomic imprinting goes back to eggs and sperm. And so basically, males and females can methylate different genes and they can turn off different genes. So let's look at these two syndromes. We have Prader-Willi syndrome and Angelman syndrome. In the same region of the DNA, you will have, in Prader-Willi syndrome, you will always have paternal silencing of a gene. So this is in the sperm. And in Angelman, you'll have maternal silencing of the gene, and this is in the egg. And this is normal. There's no problem with this at all because for Prader-Willi syndrome, this section of the gene has to be expressed on maternal chromosomes. And for Angelman syndrome, this section of the gene has to be expressed on paternal. Now, what happens for these diseases, however, is that there is a lack of expression of the gene. And so what happens is that you have the disease occurs when the gene that is in the deleted region of one chromosome is methylated on the other chromosome. So again, in Prader-Willi, the paternal is going to be off because that's been decided in the sperm. And in the resulting person, they have a methylation on the maternal gene. And so the only gene that's actually functional, the maternal gene, gets methylated and turned off. And so this leads to the actual syndrome. Now in the Angelman, it's the opposite. The maternal egg is turned off, and so you get methylation of the only other gene, which is on the paternal chromosome, and this methylation causes the paternal, which should normally always be on, to turn off. And then you get the syndrome associated with this. And because methylation is so hard to reverse, this is what leads to the actual disease. You don't have any of those proteins being made. Now finally, we're going to talk in the next about activators and co-activators. So we're going to have in eukaryotes sequences called enhancers. Enhancers are going to either be close or far away from a gene that they are going to help regulate. And so you can have various different regulatory sequences, and these sequences can help to turn on genes.